Hey science lovers, welcome back to this channel. It's going to be the first episode of a series of podcast-like videos. Today we are diving into a super cool molecular biology technique called the pull-down assay. It's not flashy, doesn't glow or spin or explode, but it tells us one of the most important things in biology, how proteins interact. And if you've ever wondered how we figure out what proteins are talking to each other inside cells, this episode is for you. So, segment one of this episode, what is a pull down assay? The pull down assay is basically a fishing expedition in a test tube. You take a bait protein, attach it to a solid support like agarose beads, and then drop it into a solution containing other proteins, the prey. If your bait binds to anything in the mix, you can pull it down out of the solution, wash away everything else, and then analyze what's stuck. It's a type of affinity purification, and it's used to study protein protein interactions, which are crucial for everything from cell signaling to immune responses. Segment 2, how it works step by step. So, here's how it usually goes down in the lab. Firstly, you have to tag your bait. The bait protein is expressed with a tag like histidine tag, GST, or flag. These tags help immobilize the protein on a resin or bead. Secondly, you have to bind the bait to a solid support. You attach the tagged bait protein to beads that recognize the tag, like glutathione agarose for GST tags. Thirdly, you have to incubate with a protein mixture. This could be a whole cell lysate or a purified solution, depending on what you are testing. The first step is washing. Non specifically bound proteins are washed away so only strong or specific interaction remain. The last step is the elution and analysis. The bound proteins are eluted and run on SDS page or analyzed by Western blot or mass spectrometry. What is the key? You are not just seeing if protein interact, you are seeing what interacts with your protein of interest under near physiological conditions. Segment 3 of this episode, why it matters and the applications. Pull down essays help us understand or answer critical questions in the biochemistry and cell biology. So the main three questions to answer is, does protein A binds to protein B? Are interactions dependent on certain modifications or conditions? What unknown partners bind to a newly discovered protein? It's often used alongside with other assays like co-immunoprecipitation or yeast hybrid to validate the interactions. So I give you one cool example. Scientists used pull-down assays to map out interactions in cancer cells, identifying protein partners involved in tumor growth. So it's a potential drug targets. They are also used in drug development to test how small molecules affect protein complexes. Segment 4 of this episode is the pros, cons, and wrap up. Like any technique, pull down essays have pros and cons. The pros are simple and relatively low cost, can be very specific if well designed, and it's good for detecting strong direct interactions. For the cons, we have also three. So it's might miss stringent or weak interactions, the interaction that may not survive to wash. Secondly, there is a risk for false positive from sticky proteins, the one that binds everything. And thirdly, it's in vitro, so it doesn't always be a perfect mirror for what is happening in the living cell. But when done right, there is super powerful, like giving you a backstage pass to the protein protein interaction concert. So that's for today. Drive into the pull down essay. Whether you are in the lab or just a bio nerd at heart, Now you know how scientists fish for molecular partnerships. Thank you.